Great. So my name is Cole Lyman, and I am a rising junior at Brigham Young University. Over the summer, I studied with Dr. Peter Krashenko, and I compared various trajectory reconstruction methods in single-cell RNA-seq data. To begin, I will move to the next slide. I will explain what single-cell RNA-seq is. Essentially, what this type of data is, it measures gene expression at a high resolution, so on a cellular level. So this is an image of one method of collecting single-cell RNA-seq, which Victor later on today will talk about. This is called the droplet method. And in each droplet, there is one single cell, as well as a set of unique barcodes that identifies the nucleic acids in that cell. And so then the nucleic acids are sequenced, and you can identify all of the characteristics of that cell. And with this advent of this new technology, there are two sets of categories of algorithms that are needed in order to analyze this data. The first being clustering, or subpopulation classification. And this essentially groups cell types that are similar together. The next being trajectory interpretation. So once you have classified and clustered these cell types, there can be a trajectory that moves from one cell type to another. So this is applicable in studies of cellular differentiation. So if you take an example of a stem cell moving into various cell fates, you can map the trajectory of how that cell progresses, as well as pseudotime measurements. You can analyze the various phases of the cell cycle and see what the differences are in gene expression. And another use for this is metabolic response. You can introduce a factor into the cells and measure in real time what the gene expression effect has on that whatever factor you're analyzing. So as most biological data goes, this data is very noisy, and the trajectories that are mapped are not always just one single line, but they have what's called branching. And this is an example of a branching trajectory. So this is where the trajectory starts, and then one moves towards this end and one moves towards this other end. So identifying these branches is critical in understanding complex biological processes such as cellular differentiation. Cells can form into various cell types and not just one other different cell type. And there are various methods that are used to identify branching in single-cell RNA-seq data. I chose two of these methods, one of which is called wishbone algorithm, and the other is Krashenko's algorithm. And it is important to note that just because you can map a trajectory, and just because it seems that there's statistical significance that there is branching in the data, doesn't mean that actually means anything in biological terms. It's important to understand your data and to make sure that the trajectories that you are mapping actually mean something. So in order to compare these algorithms, we had to select two data sets that would provide an unbiased comparison of them. We used a tool called Pagoda, which is made by Gene Fan, and this provided a statistical snapshot, so to say, of the single-cell RNA-seq data. And this is what one output of Pagoda, a Tisney plot, which Ben talked about earlier, is a two-dimensional representation of high-dimensional data. The two data sets that we chose were myeloid progenitors in mouse and mature blastocysts in human. And in the myeloid progenitors, you can see that the cell types are clustered, but the clusters are based on cell type. So each different color represents a different cell type that comes from the myeloid cells. And in the mature blastocyst data, each color represents a different embryonic day, ranging from embryonic day 3 to embryonic day 7. In order to compare these algorithms, one must understand how they work. The wishbone algorithm, it first constructs a nearest neighbor graph. And so in this diagram, you can imagine that each of these points is a cell, and each edge connecting those points in the nearest neighbor graph is a possible route of the trajectory. Following this construction, the construction of this graph, the user inputs an early cell. So this is the beginning of the trajectory. And then diffusion maps are created. And the purpose of diffusion maps are to remove short circuits in the nearest neighbor graph so that the trajectory does not circle in one part of the trajectory but can move all the way through the data. Another purpose of the diffusion maps are to reduce the dimensions of the data. And this is important to note later on when we analyze the results, that the trajectory is constructed in low dimensionality. So following the reduction of the dimensions, random waypoints are picked along the trajectory, represented by these black dots, and then the cells are grouped, and the disagreements of the groups of cells that have high levels of disagreement, so for example this green cell group and this red cell group, they have a high level of disagreement between each other. That is identified as a branching point. So one assumption that the wishbone algorithm takes is that there is only one branching point in the data. So there can be a maximum of 2 branches, and this is a limitation because in biology we see more than one branching point in data. So how Krushenko's algorithm works is it utilizes the data that is provided by Pagoda through taking that grouping of cells. So the cells are already clustered using Pagoda, and the principal points are placed at the local minima of the cells. So if you can imagine that each point represents a cell in high dimensions, each group of cells gets a principal point, and then following this principal point, there is a minimum spanning tree constructed, and this minimum spanning tree forms the trajectory. So here are the results from the myeloid progenitor data set. On the left is Krushenko's algorithm results, and on the right is Wishbone's algorithm. So I'll explain how you can interpret Wishbone's trajectory. So if you see this graph, this metric right here, the cool colors are the beginning of the trajectory and the warmer colors are the end of the trajectory. So if it were to be a line, it would start here, move this way, and then jump up over here. And on the right, this is how you can identify the branching. So it starts with blue and then moves to red and to green. And in Krushenko's algorithm, the trajectory is the line that goes throughout. So as you can see, Krushenko's algorithm has a significant branch in each of the various cell types, which is good to know because that's what we were expecting to happen. But in the Wishbone algorithm, it's a different figure that's not shown here. It was quite ineffective in clustering the correct cell types from the labels that are provided in the data. And here are the results of the mature blastocyst data set. You can see in Krushenko's algorithm that there's a very clean trajectory moving from embryonic day 3 in the correct order all the way to embryonic day 7. There's some nice branching points there. Here in the Wishbone algorithm, you can see that most of the cells are found in the early part of the trajectory, and there are very few cells that are found in the later part of the trajectory. And the branching points that it identified don't fit with any biological meaning that we can see so far. And so some reasons why we think that this happened is that Krushenko's algorithm, it uses the gene sets that Pagoda uses in order to group the cells not only with the gene expression, but also with the gene processes. So similar genes will be rinsed together, not only if they have a similar expression level. Whereas in Wishbone, it simply uses principal component analysis to group these cells together. One thing about Krashenko's algorithm is it has a very low variability, so the principal points are computed in high dimensions. So these principal points are able to encapsulate a lot of information, so they don't change very much. Whereas the wishbone algorithm uses the diffusion maps, and depending on the various diffusion maps that you choose, the trajectory can change drastically because it looks at a different dimension of the data. And so that is one weakness of the wishbone algorithm. So I would like to acknowledge everyone that helped make this summer possible: Dr. Krashenko, Jean Fan, Ruslan Soldanov, Suzanne Churchill, Barbara Mon, Dominique Torrejos, and all of my peers. So thank you very much.